our topic is gluten control in India, where we are and how we can go ahead with the current situation of the vaccination in India. So I know everyone knows about the disease, but to discuss about what I am laying points on in the coming slides, I just want to cover the basics of why blue tongue disease is important. It's a double stranded RNA virus and 10 segmented virus. So this is important because we'll know why the control of the disease is still difficult because of the biology of the virus too. So as you can see, it's a segmented double-stranded RNA virus. So segmented itself means that so the biology itself is designed in such a way that it has high recombination rates, and that can be seen in the number of serotypes that are there in the virus in India. So totally in the world, 29 different serotypes have been reported, confirmed. In every quarter, there are new articles published reporting a new serotype. But as of now, 21 known serotypes are there circulating throughout the world. And in India, we have 24 of them. So that indicates how important this disease is in India. So these 24 serotypes have been reported either through virus isolation or through serology at some point of time in India. And outbreaks can be seen of this disease every year almost, and it causes severe economic losses to the farmers. And this disease is transmitted by the bite of Culicoides mosquito in Culicoides bug. So a vector is also involved. Now, if you look at the common clinical signs of the disease, so um, as you can see, hyperemia and edema of lips and nostrils is very common with serous to mucoid nasal discharges. Lips are usually swollen with grayish brown necrotic deposition, and nasal areas show rhinitis along with mucopurulent nasal discharges and excoriations. Oral mucosa can also be, you can see oral mucosa congested and edematous. BTV affected sheep show extreme weight loss, severe lethargy, dermatitis, and break in the wool. Torticollis is also quite common, and reluctancy to move because of the necrosis of skeletal muscles is also highly seen. So hyperemia of the coronary band in the feet, that also makes the animal less mobile. And if you look at the lungs, they will be heavy, edematous, congested, and hemorrhagic, but not collapsed. So these are the common symptoms, and when you look at the common postmortem lesions, this is what we'll see. And now I'm not going into details of exactly what this disease does in a host. We all have studied it, and I just want to refresh the memory because how it causes the way what clinical signs we see, because they vary. It depends on how the virus is actually, what the virus is actually doing. Not all serotypes are equally virulent. So depending on how pathogenic a serotype is, we'll see the clinical signs and the pathogenic the lesions also. Now, what happens after the bite of the Culicoides species in the ruminants is the virus is transported by the dendritic cells of skin to regional lymph nodes. That is where the primary replication occurs. And then from there, the virus reaches the blood circulation and then spread to the different secondary organs where the virus replicates in endothelium and mononuclear phagocytes. So as a result of this extensive endothelial damage, there is a release of cytokines in excessive levels, which we call it as cytokine storm. And that is what is responsible for the pathogenesis of BT, like increased vascular permeability, severe hemorrhages, edema, effusions, thrombosis, intravascular coagulation, and everything. So if BTV is seen in pregnant animals, it can result in the cerebral malformations in the offspring, and also some abortions if found in the last stages of pregnancy. Now the diagnosis, coming to the diagnosis, as I said, the disease outcome ranges, there is a wide range of the disease outcome. It can sometimes be inapparent as well as to the fatal stage. Again, that depends on the type of the virus. Why the type of virus means it depends on the serotype of the virus. So depending on the which serotype is most prevalent in a particular season, the disease symptoms also vary. Also, it depends on the host. So Deccanese sheep are much more resistant, or much more, the clinical signs are not as apparent as in Nellore sheep. And also the environment and the in stress conditions, they also play a role in the clinical outcome of the disease. But the commonly found clinical signs would be pyrexia up to 42 degrees, excessive salivation, depression, and panting. And initially, we'll see clear nasal discharges, which they later become to mucopurulent, and they may dry and form a crust around the nares. And hyperemia and congestion of the muzzle, lips, face, eyelids, which can cause edema. Ulceration and necrosis of the mucosa of the mouth. The tongue may become hyperemic and edematous and cyanotic in later stages and can protrude from the mouth, which gives the name of the disease, the blue tongue, but that we rarely see, especially in Indian breeds. And also extension of hyperemia to the coronary band of the hoof and causing lameness. Torticollis in severe cases, and abortion or either birth of malformed lambs. Complications of pneumonia, and in severe cases, death can be seen in 8 to 10 days. If not, there is a long recovery, alopecia, sterility, and growth delay, which affects the meat farmers a lot. So the lesions, postmortem lesions, as I have mentioned in the previous slide, there will be congestion, edema, hemorrhages, and ulceration of the digestive and respiratory tract. There is severe bilateral broncholobular pneumonia. And in fatal cases, lungs may show intraalveolar hyperemia, several alveolar edema, and bronchial tree may be filled with froth. Thoracic cavity and pericardial sac may contain large quantities of fluid. Distinctive hemorrhages will be found at the base of pulmonary artery. There will be congestion of hoof, laminae, and coronary band, and then hypertrophy of lymph nodes and splenomegaly. So, differential diagnosis, again, these most of these symptoms are also seen in other diseases, especially contagious ecthyma, FMD in sheep, plant poisonings, photosensitization, parainfluenza 3, pneumonia, pestis, PPR, epizootic hemorrhagic disease of deer, vesicular stomatitis, foot rot, especially foot rot, because people both occur in the same season, and both in both the conditions, sheep is very hardly, it reluctantly moves. So people confuse foot rot with blue tongue or blue tongue with foot rot. Obviously, sheeppox and infectious bovine rhinotracheitis also. So these are the things that we have to be careful while differential diagnosing. Now coming to the prevalence of the disease in India. So this particular image shows how prevalent this disease in India. As you can see, almost all the states can be seen with the disease, either by seroprevalence or by virus isolation. But if you look closely at the map, at the southern states of India especially, it is very high in both sheep and goat. Also, cattle seroprevalence in sentinel animals, that is also very apparent. So that indicates the importance of the disease in southern India and why we should control it. I mean, southern, especially AP and Telangana, rank top in the sheep and goat population in the country. And now to control the disease, again, all viral diseases, it's better to control them by vaccination. That is what we have been following all this while. And I cannot proceed with this talk further without mentioning the importance of ICR Network Project Blue Tongue, which was initiated in 2002. That was when this was taken up seriously, because till then there were very few isolates known in India. Even when they were done, isolation was done, it was in isolated parts of the country with no network, so they were not sharing. It was not possible to know who has what isolates. So Government of India along with ICR had formed this network program on blue tongue, and they have centers throughout the country, starting from Hisar IVRI to Tanuvas, Kerala, AP, Angru, then in Maharashtra, I believe, and then Madhya Pradesh. So there were a lot of centers to begin with, but they didn't, Tamil Nadu also. And not all of the centers were maintained till the end of the program, but this program has initiated enormous efforts in the isolation of blue tongue virus in the country. And because of this program, the first vaccine in the country was also launched, inactivated vaccine, which contains 5 different serotypes: 1, BTV1, 2, 10, 16, and 23. So these isolates were isolated, and the technology was transferred to many companies, including IIL from Dr. Bhaskar is a part of it, and they manufacture Raksha Blue vaccine, which is extensively used in AP and Telangana states. And then to several other companies also, this vaccine is commercialized. So now it is already there. So why do we need an advanced vaccine? So there have been certain reports from the field that despite being vaccinated, we are still seeing outbreaks. And in some flocks, it is protecting very well. And in some seasons, it is protecting very well. But then in certain flocks, it is not. Again, it depends on a lot of factors, like when actually we are vaccinating. So one problem that I have noticed when I talk to the sheep farmers is that once they hear about the outbreak in the neighboring farm, they tend to vaccinate then. But by then, the virus must have already been spread to that particular farm, and there is no point in vaccinating then. So before the initiation of monsoon season, because it is spread by a mosquito, Culicoides, it is always preferable to vaccinate the animals right before the monsoon season starts. But due to many reasons, that if that is not possible, even if the vaccination is done later, and once the virus is infected and if you're vaccinating in the incubation period, that will obviously not be as effective as vaccinating before the spread of the disease. So that is a technical part of it. Now coming to the biological part of it, as I have said, the vaccine contains the 5 different serotypes, which is 1, 2, 10, 16, and 23, because those were isolated in the initial phases of the network project, and those were very much prevalent then at that point of time. And as I said, India has at least 24 different serotypes being circulated at different points of time. So that could be the reason. Now, the question is, now the important point to note here is the serotypes are not cross-protective, which means if even if they are, there is very limited cross protection, which I will address in the coming slides. How much cross protection is there between the serotypes, but much of the cross protectivity is very limited. So that could be another reason, and which is what but then we cannot keep on changing the vaccine serotypes every season that is coming, because it needs a lot of official permissions and everything. It's a long process. So how can we address this issue? That is where we initiated. That is where we started working. Again, as I said in my previous slide, if you the blue bars indicates the time period between 2014 to 16, the orange bar indicates the time period from 2016 to 18. If you look at the prevalence of the different blue tongue serotypes, if you look at BTV1 during 14-16, it was very good percentage. 16-18, it is very low. Likewise, BTV2 in the initial phases, in the initial 3 years, it is very low. But then you can see that it went up in the 16-18 period. As you can see, there is a lot of variation of the blue tongue viral serotype circulation between the seasons, or between among the different years. Like BTV21, it was not there in 14-16, but you can see it coming up. BTV23, it was there in 14-16, totally not there in 16-18. So you can see there is a lot of serotype variation circulation in different years. And so again, I want to mention the project that we, after network project ended, or when it was towards its completion, we started another project funded by DBT and BBSRC in UK, because it's a big problem there as well. We started a collaborative project, and then that is when we did this circulation study, like how exactly it is being circulated. We used a lot of advanced tools. Before that, it was BTV serotype prevalence was checked by Zero SNT. Now we have standardized real-time PCR, all these latest molecular biology techniques. Isolation was done at a faster pace, and diagnosis is done at a faster pace, and we were able to see this variation because of that. And also this is seen in 2025, the latest serotype prevalence. As you can see, there is a lot of variation. Now, one thing I want to emphasize here is, if you look at BTV4, this is consistently been seen rising from 2014 to 2025. However, the vaccine does not have this serotype, BTV4. And BTV24 is also consistently rising up. 2 is there, and good thing the vaccine also has BTV2. And one more important thing that we have noticed is, as you can see in the later part of the graphs, there are a lot of mixed infections. That means from the same animal, when we isolated BTV, it belonged to 2 or more than 2 different serotypes. As you can see in the end, where this particular animal, it had both, not both, it had 2, 4, and 21, all 3 different serotypes simultaneously in that particular animal at that point of time. So this is a big problem, because as I said, 24 different serotypes in India. So to address this issue, so now we can see 2 major reasons why the vaccine could not be performing as effectively as it should be. One is this variation in serotypes. That is the major thing that we want to address. So if you want, and then we wanted to get into developing this new generation vaccines. So now we all know the COVID has given us new generation. Now everyone knows about mRNA vaccines, subunit vaccines, recombinant vaccines, thanks to COVID. So it has been there since before, but then COVID made it really popular. So that is how we wanted to address this issue by developing a recombinant vaccine. The idea there is, is it possible to develop a single vaccine which can protect it against multiple serotypes, because the serotypes seem to be evolving. So we cannot keep changing the serotypes in the vaccine. So why can't we just design one vaccine which is good for many of the serotypes? With that objective in mind, we have initiated this study. And as you can, to understand the biology of the virus, I'm not going into detail of it. If you can see this outer surface proteins, which are VP2 and VP5. So if you want to develop a recombinant vaccine, the outer surface proteins are the most important, because that is what the host immune system sees in the virus. So these are the highly immunogenic proteins of a virus, just like the spike protein of COVID virus, which is the outer surface protein. And that is what is used in the recombinant mRNA vaccine, that Covishield. Just like that, we wanted to go with the outer surface protein of the blue tongue virus and see if these are good enough to be used as a recombinant vaccine. And now the problem with using VP2 was it is highly variable. Let's say there are 29 different serotypes of blue tongue virus. The VP2 of each of those serotypes is completely different. So we cannot make one VP2 and then expect it to work for all the serotypes again. So with that in mind, we have identified a conserved region between 338 to 383 nucleotides, amino acids, sorry, in this particular VP2 protein, and hope that maybe this conserved region among all the serotypes, if we use that part as a vaccine, maybe that can protect against multiple serotypes. But since this is a very small part of the VP2, you cannot expect that 50 amino acid protein can protect against the whole disease. So we have also used whole VP5 protein, because that is also an outer surface protein. It is not as variable also. So we have taken a part of VP2, which is identical in all the serotypes, and the whole VP5, which is also identical in all the serotypes. So these two we have used. And then before we go ahead and make this a protein and then give the animals this and check it, now there are a lot of tools in silico in a computer. We can check whether this is going to be good enough as a vaccine or not. Is this protein immunogenic or not? So we have checked that, and this particular protein, the conserved part of the VP2, seem to be very good immunogenic with B cell and T cell binding epitopes in it, which are what are important for a vaccine to be effective. And then we went ahead and cloned, expressed, and purified three different proteins, that is CVP2, VP5, and NS1 also. NS1 because it is a good T cell promoter, T cell activator. So we have all these three proteins of blue tongue virus we have made into recombinant proteins. And then first we have checked the antibody production in mice. So we infected mice with these proteins, collected blood at time points, and then we have checked whether antibodies are being generated with these proteins or not. And they are being generated. As you can see, both VP2, VP5, and NS1, they have produced very good antibodies against these proteins. And now the next thing was to check whether the same is being replicated in sheep, because sheep is a natural host. So we have done that again, individual proteins, and then together also into sheep. And then it's a 60-day study. At frequently time points, we have collected the blood and checked whether it is generating antibodies or not. As you can see, they are generating antibodies. This is the control animal. The black group is the control animal. And what this graph shows is all these proteins are generating a good immune response in sheep as well. Now coming to the final question, which is are the antibodies generated because of these proteins are cross protective or not? Because that is the objective for this, right? We wanted to generate a single vaccine which can protect against multiple different serotypes. So what we did was, after we've collected the serum from the CVP2 and VP5 group, we have isolates in our lab, BTV 1, 2, 4, 5, 9, 10, 12, 16, 21, 23, and 24. We have these different serotypes in our lab. What we used was, what we did then was, we tried to neutralize all these serotypes with that single serum sample, which has antibodies against CVP2 and VP5. So if that was protecting, if that was working as a broad spectrum vaccine, it should neutralize many of these serotypes. So as you can see the numbers, numbers indicate good neutralizing antibody response. I just want to highlight that BTB 5, 12, and 24, except for these three serotypes, all these serotypes were neutralized by this CVP2. Now then we tried it multiple times, still these three serotypes could not be neutralized. So we wanted to see what exactly is happening. So this particular image shows the relation of different BTB serotypes among themselves. As you can see, the number indicates the number of serotype. So BTB 24, as you can, so thicker the line, greater the cross protectivity is. So 20 and 17 are very good related. So 17 can cross protect against 20 also, which can also cross protect against 4. But 4 and 12 are not as good related. So you can see the dotted line. So it won't protect. I mean, antibodies against 4 will not protect against 12. Likewise, if you see 24, it has nothing. I mean, it is not related to any other serotype, which could explain it's extremely high variability, and which could explain why we were not able to neutralize 24 with the cross-protective CVP2 vaccine also. Also, 5 is very weakly related to other serotypes. Maybe that could be the reason. This is what we have thought about. So to summarize our findings so far, we have generated these recombinant proteins using different, again, this was not easy. We had to try different conditions, and we have generated the proteins, and then we have checked that those are good enough immunogenic and generated neutralizing antibodies. And this was the first report of the conserved part of the VP2 segment and its immunogenicity in sheep, and high neutralizing antibody titers were observed. Now what we are doing is we just want to minimize the amount of effort that goes into administering a recombinant vaccine. So we are trying to, the top part is the conserved VP2 and the bottom part is the whole VP5. Instead of giving them as two different proteins, we try to put them together, stitch it together as a single protein and give it. So this is what the protein's structure will be. If we stitch the CVP2 and VP5 together as a single protein, it will also work as a DIVA vaccine against BT, because we are using only two proteins. It can differentiate the vaccinated and infected animals too. And as you can see, the many numbers that you see on in the left side, the many numbers, these indicate the immunogenic epitopes present on this protein. That means these can be very good as viral candidates, vaccine candidates, because they can generate a very good immune response when administered as a vaccine. We have tried different linkers, to link the two proteins. We have tried different linkers, and we have shortlisted this particular one, KP12. So that's seemed better with the with the immunoanalysis in silico. We found that this is better than the other four. So we have shortlisted that, and now we are in the process of generating this particular chimeric vaccine, and then again it will be tested in sheep like before. And then after, so what we'll be looking for there is, again, it's cross protection ability, because that is what the whole point of doing this study. So we'll have one vaccine which can protect against multiple serotypes, because blue tongue seems to be evolving rapidly in every season, and new serotypes seem to be circulating every season, every other season. So we cannot go back and keep on changing the virus. I mean, you just design something now and then next season something else comes up. You cannot just keep on editing the same vaccine again and again. So we have used an immunoinformatics approach to design the subunit vaccine, and which has protect good humoral and cellular immune responsibilities, and high neutralizing antibodies were identified. Now I have come across this particular risk map prediction strategy. This is a work done by Nivedi, Dr. Mudassar Chanda's lab. So this is very interesting. Now this particular study, what they have done is they have made some used some tools, very advanced tools, which I am not an expert of, but they have used a lot of tools to predict the disease. So for that, as you can see, the first point, they have used data. blue tongue disease data from 1997 to 2011 in South India, and that data they have used, and they have used different models, bioinformatic models, to see which kind of environment, which kind of host factors, which kind of vector factors are all involved in predicting a disease. So this again emphasizes the importance of accurate disease reporting, because without correct disease reporting, these kind of studies cannot be done. So as you can see, they have used that, and then they have predicted the disease. As you can see, states of AP and Telangana, along with Karnataka and Tamil Nadu, you can see them blazing red, indicating that, these three are very high risk factors. So this can be done in every season. So the environmental factors can be put in that model, and the BT presence and absence can be put in that model. The host, what kind of breeds are prevalent, what kind of age group, what kind of culicoid species are prevalent. So everything can be put in the model, and it can predict the disease prevalence, or we can predict the disease in the coming seasons. I think these kind of strategies are very, very much important for us, because if only by combining all of these strategies, we can be able to uh, control the disease better. I mean, all of us have to, it should be a combined effort. Field veterinarians, policy makers, research scientists, all of us, I think all of this is a combined effort, and only with this combination we can be able to ultimately cater to the farmers by controlling this disease better. Thank you so much for listening.